Hey everybody, Mr. Montali coming to you live here. Our first distant learning topic will be reproductive cell formation. You'll have to bear with me, it's actually pretty tough speaking to a screen as opposed to 30 kids. So we're going to try and do this in one fell swoop. And if there's any questions on the topic, I'd be more than happy to answer them during our meetup sessions per block. Meiosis also can be found in the e-text chapter 11 section 4, and it literally is a way to make reproductive cells, sperm cells and egg cells. Two major cell types, cells that are regarded as diploid or diploid, which have two sets of chromosomes, and the types of cells that would be regarded as diploid are all somatic, also known as body cells, brain cells, muscle cells, skin cells, and so forth. Little picture of some skin cells there. So these would be cells that essentially have two full sets of chromosomes. These chromosomes would be homologous, meaning they are alike but not identical to one another, and we'll get into that a little bit later on. Why do we have two sets of these chromosomes per cell? Well, you should know that obviously from the law of segregation and our genetics section. Two parents obviously are going to donate a set each, so a new living organism would get two sets of chromosomes because they get one set from each parent. Now, we have another type of cell, a cell that will really be more focused on in this particular topic, and they are haploid cells. Sperm cells are haploid, and egg cells are also haploid. These types of cells have one set of chromosomes, and the process of meiosis literally takes these special cells that are designated to become mature sperm or egg cells and puts them through a process where it cuts the number of chromosomes they have in half. So gametes, the sperm cells and the egg cells, don't have two sets of chromosomes. They have only one set of chromosomes. So essentially when we start with this process, we have an immature sperm or egg that needs to go through a process of maturation and reduction of chromosomes to become an actual reproductive cell. So we're going to go from a simple diploid cell into multiple genetically different haploid cells. That's a lot, and we'll get into it more as we progress through this PowerPoint. Let's talk a little bit about homologous chromosomes and what it means to be homologous. I'm going to show you a karyotype here. This is a karyotype that's been digitally enhanced, and you can see that it does have two X chromosomes, so this would be of a female gender. And you can see that each spot, 1, 2, 3, 4, all the way to 23, have two chromosomes per number. Well, each of those chromosomes are homologous. Now, in the next diagram, you can see that these particular chromosomes haven't gone through a replication process, so they're single-stranded, if you will. And you can see that although they are different, because one came from the mother and one came from the father, they code for the same genes or the same characteristics. And you can see the characteristics are represented by the alleles R, P, A, B, and C. Again, things that we covered when we talked about genetics, or we wrapped up our genetics section. Okay. Pairs, homologous chromosomes are pairs of chromosomes that are found in diploid cells. You wouldn't find homologous chromosome pairs in reproductive cells because they only have one set. So nonetheless, homologous chromosomes are found in diploid cells. And once again, homologous chromosome pairs are alike in the alleles they carry, but they're different because they were donated to you or a daisy or a fruit fly or a Patricia poison arrow frog from different parents. Okay, so each of the chromosomes within a homologous chromosome pair are different because they've come from different individual parents, but they're alike in the fact that they code for the same traits. So that's a little bit about homologous chromosomes, and they'll come into play quite a bit in our next few segments. Okay. Just as a visual, we see a couple of pictures here. Chromosomes prior to replication and then after. I have A1 and A2. Excuse me. A1 and A2 represent a given allele for a characteristic. And you can see prior to replication, we have 2 chromosomes. They are homologous. A1 and A2 code for the same trait. Okay, so we might say that that particular allele is the allele for hairline. Now, when we look to our homologous chromosomes after replication, each of those chromatids have doubled, and we know that the alleles from A1 are identical and the alleles from A2 are identical. Now, are A1 and A2 identical themselves? No, because they've come from 2 different parents, but they do code for the same traits, which would be, in this case, we said were hairline, either straight or widow's peak. So that's kind of a visual representation of homologous chromosomes and the alleles you would find on them. So hopefully you realize at this point, we know that A1 and A2 are the allele that codes for the same trait. In this case, we said it was hairline. Okay. But what do we know about the alleles labeled A1 or A2? We know that the A1s on the green chromosomes are not going to be identical to the A2s on the purple chromosomes. We may have, the allele for straight hairline and widow's peak, a heterozygous scenario on the A1 and something different on A2. So nonetheless, homologous chromosome pairs, the chromosomes are alike in what they code for, but they are different in regards to having come from different organisms, parents, if you will. Meiosis, the actual process itself. It literally is a cell division process that produces reproductive cells. That's all it does. And it's going to sound similar to mitosis. It's going to have the similar stages, but it does a very different end result, or it gives you a very different end result. The process of meiosis makes reproductive cells. Sperm cells, egg cells. The process begins with one immature gamete. It doesn't begin with just any cell. The cells that become sperm cells or egg cells are the product of a special cell designed to go through a maturation process and become a mature gamete. These are called spermatocytes for sperm cells or oocytes for egg cells. And they start out carrying the diploid number of chromosomes, which would be different in all different types of organisms. If we were talking about a human being, the spermatocyte or oocyte would start with 46 chromosomes. If we were talking about a fruit fly, they would start with 8 chromosomes. So depending on the organism, it's going to have a different diploid number. The process ends with 4 genetically different gametes. So you start with one diploid cell, and your end result are 4 genetically different sperm cells or egg cells that have the haploid number of chromosomes. So if we were talking about a human being, we're going to begin with a spermatocyte or oocyte that has 46 chromosomes and eventually work our way through the process to the end game, which would be 4 cells that are genetically different that have the haploid number. So in humans, you'd start with a cell of 46 and eventually have 4 cells that each have 23 chromosomes. You're familiar with prophase, metaphase, anaphase, telophase from our talks of mitosis, and these stages are used as well in the process of meiosis. Instead of just one round of PMAT, though, it goes through 2 rounds, which we'll talk about shortly. It all begins with meiosis 1. So meiosis happens in 2 stages, meiosis 1 and meiosis 2. Meiosis 1 has a special name. It's called reduction division, because after the end of meiosis 1, we'll have reduced the chromosome number in half. So if we're talking about a human cell, our spermatocyte or oocyte would begin in meiosis 1 with 46 chromosomes, and at the completion of meiosis 1, we would have 2 cells that each have 23 chromosomes. Now, meiosis 2 does not do that. It will not reduce the chromosome number down anymore. So again, meiosis 1, also known as reduction division, because we're going to cut down the number of chromosomes in half. The stages, exactly the same as mitosis, different end result. We start an interphase where the chromosomes are replicating and the cell's growing. It's kind of doing its thing, getting prepared to go through the process of meiosis. Prophase 1, we have homologous chromosomes in a cell, and they form tetrads. All right. Now, as you can see in my burning flame there, I'm showing a pair of homologous chromosomes and the process of crossing over, which we'll talk about kind of as we go along in this presentation. Okay, so again, prophase 1, the genetic material is going to condense into these nice chromosomes, and they are going to pair up with their partners. So if we're talking about a human being, then each of the 23 pairs of chromosomes pair up and they get right next to each other. Okay. Metaphase 1 begins, and our homologous chromosomes align at the center of the cell. Okay. Just like in mitosis, metaphase middle, we're talking about the homologous chromosomes aligning at the center of the cell. Anaphase, A for apart. The homologous chromosomes move apart to opposite ends of the cell. And then finally, we have telophase, where cytokinesis will end, and we'll end up with 2 cells that have half the number of chromosomes that we began with in prophase. Now, this is a process that I will be drawing and posting in the near future. I'm just trying to kind of get the ideas out now, and then we'll post the actual drawing of how this happens in a little bit. That's the end of meiosis 1. Meiosis 2 will kick in, okay, and the stages are going to be your basic PMAT, prophase, metaphase, anaphase, telophase. There is no interphase in between meiosis 1 and 2. Prophase 2, the chromosomes condense up again. And now again, we don't have homologous chromosomes anymore. Okay? What we have are sister chromatids. Okay? The homologous chromosomes were broken up into 2 separate cells. So now we have our sister chromatids, and they're going to align at the center of the cell, just like in metaphase 1. Always something aligning in the center of the cell in metaphase. Anaphase 2, those sister chromatids are going to be pulled apart and move apart to opposite ends of the cell. And then lastly, we have telophase 2, which cytokinesis ends, and our end result is 4 genetically different mature gametes that have been produced. Okay. Again, every single sperm cell or egg cell is genetically different from all of the other ones, and that's done through the random aligning and pulling apart of these chromosomes, and also from the process of crossing over. I will go into crossing over again at a later time. It is important because crossing over creates massive amounts of genetic variation. And as we know, in the world of evolution and mutation, and adaptation. Variation is very, very important to a species survival. Okay, now, wanted to talk a little bit about the differences between meiosis and mitosis. So I just put together a little table here. Meiosis begins with one diploid cell and eventually works its way to 4 haploid cells. Okay. In mitosis, you begin with one diploid cell, and it literally creates 2 diploid cells that are identical to one another and identical to the original parent. Okay. Meiosis produces cells that are all genetically different. Variation is a great thing. Meiosis creates reproductive cells that are genetically different from all of the other reproductive cells that exist. And mitosis produces cells that are genetically identical. They're basically creating clones, just copies of themselves. But we always have to go back to what types of cells are being created. Meiosis creates reproductive cells, period. Gametes, which would be the sperm cells for males and egg cells for females. Mitosis creates somatic or body cells, skin cells, brain cells, muscle cells, blood cells, and so forth. Cells that get damaged and die and need to be replaced. Literally, it's just a factory to create a process that creates copies of cells to replace ones that are dead or that are damaged. Okay. Meiosis would be a cell division method needed by organisms that reproduce sexually. If you reproduce sexually, you produce sperm cells and or egg cells, or in some organisms, like we talked about with the pea plant, both. Mitosis is a cell division method needed to replace, that should say dead, it says deer, I'll have to change that up, or damaged cells, dead or damaged cells, and also it's a way that living organisms grow. Our cells don't just get bigger, we add new cells to grow. When you look at your baby boxer puppy at 2 weeks old, tiny. You look at them at 2 years old, very big. It's added a lot of cells through the process of mitosis and cytokinesis. Now, I'd like you to ponder a couple of questions, and I will have a submission site for you to upload your answer to this. Couple of things. A Florida Panther, who I believe will win March Mammal Madness, spermatocyte with the diploid number of 38 goes through the entire meiotic division process. So it's going through meiosis. How many cells are the end result, and how many chromosomes does each cell have? Okay, so basically, you're going to be thinking about one spermatocyte from a Florida Panther going through the entire process of meiosis. What's the end result in regards to the number of cells, and how many chromosomes does each cell have? Now we have another variation of a Florida Panther there, obviously a mutation with a dark colored coat. The second question to ponder would be a Florida Panther skin cell with the diploid number of 38 goes through the entire mitotic division process, so it's going through mitosis. How many cells are the end result of mitosis and obviously cytokinesis, and how many chromosomes does each cell have? Obviously, the answers to 1 and 2 are going to be different. Very important. This kind of is the culmination of the cell division processes, and really kind of give a great idea of whether you understand this or not. That's the deal. I'm going to have to go back and look at this and see how it sounds. Really missing everybody. Would love to be back in school and doing this the old school way of discussion. But nonetheless, if you have any questions on this, let's meet up during the meetup and do our thing. All right, guys. Appreciate it. Be safe. Wash your hands.